welcome everybody for this evening exploration of regeneration. So a number of you, when you think of regeneration, probably think of all the things in your body you wish you could regrow, particularly people of a certain age, such as myself. But of course, through history, regeneration has been viewed with different lenses. And inevitably, actually, it's been viewed by the ancients as a sign of the gods or a sign of something gone wrong. It hasn't been viewed very positively. So here in this opening picture, you see our archetypal regeneration biologist, Hercules, with one of the regenerating systems he's chose to study, which is the Lernaean hydra. And the Lernaean hydra has the rather unfortunate characteristic that if you lop off one of its heads, two come back. And actually, there's one head that you can't actually lop off with anything. So it has quite the ability, but it's quite amazing in this story that the way that Hercules defeats the regeneration of the Lernaean hydra is by cauterizing what we'd call the blastema of the healing head. And so actually, even the ancients probably had a fair understanding about the cell biology, or at least at a rudimentary level, of the way that regeneration happens, and they projected it into their myths. So this is a rather fearsome creature that took a lot of cunning and brute force to defeat. But the hydras that we study in the lab these days are nowhere near as fearsome in size or scale. This is an invertebrate hydra system. And when we talk about regeneration, what we really try to do in the lab is recapitulate what this system is able to do. A hydra has different cell types in its body. It has a top and a bottom, and it has specializations of cell types. But no matter how you dice it and cut it, it can regrow them all. So really, that's basically the principles of regeneration science, that we want to be able to take individual cells and sometimes single cells and regrow them to all the cells of the body of the system that we're studying. So that's as simple as it gets as an explanation of what we mean about regeneration. And it gets a bit tricky, and I'll wade into some of the ethics and I'll wade into some of the challenges just to tell you where we are within the science as we move along. So probably some of you know that our skin is a fairly regenerative system. Probably some of you realize the liver is fairly regenerative. Thank God for that. And the system that I study, skeletal muscle, it's also regenerative. And however, there's a general trend, although it's not absolute, that in lower vertebrate systems or non-mammalian vertebrate systems, there's a regenerative capacity that's greater. So think of the lizard and its tail, and zebrafish, there's a large number of regenerating organs. But then as we move up to more complex mammalian systems, we can no longer regenerate our brains or our hearts. This is a difficulty. So one of the greatest mysteries in regeneration biology is why some animal systems and some tissues can regenerate and others can't. So a lot of us try to understand that question by studying systems such as zebrafish that have a high regenerative capacity in their tissues. So that's another incarnation of regeneration biology. And then there's the idea of regenerative medicine. Can we harness the regenerative capacity of our body to replace and restore organs? And the sort of holy grail panacea view of regenerative medicine is it doesn't really matter what's wrong. If you've got a damaged tissue or an organ, we should be able to take a cell, regrow it in a dish or stimulate it in vivo and restore that tissue back to its integrity. And so it's not, it's often viewed as a really important and futuristic medicine. But I'm here to sort of give you a message that we are making headway with this, and there is actually medicine that is occurring through regenerative medicine. Most people equate regenerative medicine with stem cells. That's actually not entirely true. They're just one tool in the kit bag that we use to try and stimulate and provide pro-regenerative properties to tissues. So that's sort of a preamble. The other thing is it's actually a huge business. These slides are rather out of date. In 2020, it was supposedly worth $67 billion as an industry. It's way past that now. And so people are directly investing in the science of stem cells and regenerative medicine, and it's also now commercializable as therapies and treatments. So yeah, we've got regeneration biology in one arm, we've got what regenerative medicine can deliver and the fact that it's an industry in its own right at the moment. And all these things are coming together into an institute such as mine, such as that we can harness these properties and these abilities for therapeutic gain. So what are we going to cover today? It's a big field. It's a growing field. It's as I said, it has a huge impact across the board. What can we cover in an hour? Well, I'll talk a little bit about stem cells and regenerative medicine because I think it's important to realize that there are oils ain't oils, as Sol used to say. Stem cells ain't stem cells. There's all different types of stem cells, and what do we actually mean? So we'll have a little discussion about that so you can bore your loved ones with no, that's not the way to do it, or this isn't the right stem cell. Professor Currie told me such and such. And then I'm going to talk about my fascination, which is understanding regeneration biology and regenerating systems. And I'll illustrate two of the systems we use, zebrafish and axolotl, and why. And then I'll talk about this fascinating system of tissue engineering where we're combining engineering and stem cell and regenerative biology in more methodological and treatment-facing approaches. Okay, so everyone's heard about a stem cell. You know, people go jet off everywhere, inject stem cells all over their body, mainly with scant knowledge or hope of any effect. And what are they exactly? And what can we use them to treat them for? And as I said, stem cells are being postulated for treatments for neurological conditions, for spinal cord injury, for burns, you name it. This is what the promise of stem cell therapy is. If you can harness the capacity of a stem cell and turn it into a tissue and organ and have it engraft back into your body, the hope is it can repair. So that's the general principle. So what are stem cells exactly? So a stem cell is simply a cell that can replicate itself and maintain something, this magical property called self-renewal. So it can remake itself, and it can also give rise to a cell that can make another cell type. Sometimes it can make a number of different cell types. It means it's pluripotent. Sometimes it can make every cell type, such as the first cell in the human embryo, then it's called totipotent. Sometimes it can only make one tissue, such as muscle, and that's a unipotent stem cell. So, but the basic feature of the stem cell is it can make a copy of itself as a stem cell, self-renewal, and it can make a progenitor that gives rise to tissue. Now, there are different types of stem cells, and we'll go into those, but the ones that I study are called somatic stem cells. So scattered through some of your tissues, not the brain so much in human, but teeth have stem cells, skeletal muscle have stem cells, certainly in the blood. There are stem cells called hematopoietic stem cells. The gut has stem cells, the skin has stem cells, and after decades of research, we now know the heart has none. That was a highly controversial organ, and it does not have tissue-resident stem cells. So some of our tissues have stem cells, and most of them can only make that tissue, right? So they're the sort of the homeostatic or repair systems with individual tissues. All right, so as I said, there are three flavors of stem cells that I'll talk about in some detail, just so we're all on the same page. So I've said the defining characteristic of a stem cell is that it can self-renew, make something of itself and differentiate. Stem cells can enable the body to grow and repair. And there are three main types of stem cells that I will call, we've got a dark horse of a stem cell called the mesenchymal stem cell. So I'll go through a little bit about that. It has a good side and a bad side, but these are sort of the professional stem cell systems that we study. So tissue stem cells, they're set aside through your development to reside in tissues and organs and are called on through injury. So most famously, our most famous stem cell treatment is a hematopoietic stem cell engraftment or bone marrow engraftment. They can make the entire blood system from a single cell. That's an amazing property. Within the early embryo, there are stem cells that can give rise to every cell of the body, as I've said, and these are the totipotent stem cells. And we used to have to study these cells by taking them from human embryos. So they were controversial because they were derived from terminated fetuses, and therefore people who had ethical problems with that did not necessarily like embryonic stem cells. They are still very good tools because they tend to be more stable and characterized than other human stem cell totipotent lines, but they are becoming less used because Shinji Yamanaka got the Nobel Prize for this process of turning any stem cell, any cell in your body into a cell like an embryonic stem cell. And these are called induced pluripotent stem cells. And these are the templates have really revolutionized regenerative medicine. So they're the three sort of stem cells that people talk about, and it just sets the benchmark for what we use. So mesenchymal stem cells. There are some cells that are derived from different areas of the body, such as fat, or even in the bone marrow, the stromal cells in the bone marrow, that can make other cell types and can self-renew. And they can influence in the endogenous niche bone repair. They're kind of bone stem cells mostly, but they are found in different flavors around the body. So they are often talked about because in the body they have stem cell functions, but they're often harvested from the body and promoted as stem cell treatments when in actual fact they don't ever restore or replace damaged tissue and organs. They do have potential as therapies because they're immune modulating cells. So they are used in conditions where inflammatory conditions, so famously you can go get a stem cell injection into your knee, but it's not replacing the damaged tissue of your knee, it's just immunomodulating your knee so it doesn't get so inflamed. There are many, many clinical trials using MSCs for different reasons, and because they're what's called an autologous treatment, they can be taken out of your body and put back into your body with very little regulation. And so therefore, they have been taken up by a lot of different practitioners who promote them. And what I learned about medicine, which shocked me entirely, was you actually don't need any evidence for something working to prescribe it. You just have to believe it may work and believe it will cause no harm. So that's actually a fairly low bar for a doctor to start a stem cell clinic, and that's exactly what's happened. So around the world, most of the stem cell clinics you will see are those people peddling, in some case, some quite rightly after clinical trials, treating patients with mesenchymal stem cells. And it's a big business in the US. Every second commercial you see on cable television is for a stem cell clinic around the corner. It's a bit like the cosmetic. I hope there's no cosmetic surgeons on the call. A little bit unregulated like cosmetic surgeries for medicine. And often it's cosmetic surgeons who do stem cell treatments. So this has sort of been a big headache for the stem cell community because they often don't do anything. And sometimes they're downright harmful if they're surgically inserted in the wrong way. That's not to say that they can't be treatments, and they are treatments in some instances, but it's a buyer beware situation for these things, right? Okay, so there are lots of clinics everywhere. I'm just going to talk about embryonic induced pluripotent stem cells a bit and what we use them for. So where do they come from? So this is a human embryo, and they come from a structure called the inner cell mass. So this is the blastocyst. This is you when you were young. And you know, it's not very dramatic, but nevertheless, this first decision to make these embryonic stem cells on which the entirety of your own tissues will be born is a very early one, and the rest forms the trifectoderm, which will implant and become into the placenta. So these embryonic stem cell lines come from the early embryo, and this is why in the US they were so controversial, why they had the act where they couldn't be used in certain states, and it was overruled in California, and that's why the California Institute in Stem Cell Regenerative Medicine was formed in order to get around that. So, but you can take these out and grow them into dish, and you can make tissues and organs. But from a therapy perspective, they're not that useful unless they're matched to a haplotype, like a bone marrow engraftment, because they're not your cells and they'll never be you. So they'll never completely match who you are. So the tissues that you make from them will always need immunomodulation in a similar way to a transplant. But they're an extraordinarily useful tools because they're very stable cell lines and good for science. Induced pluripotent stem cells are a very important step forward, as I said. They're basically initially genetically engineered cells that take particular factors, they're called the Yamanaka factors, the 4 transcription factors, that when supplied to any cell in your body can induce it to go back in time to when it was a blastocyst. It's sort of like getting in a Doctor Who TARDIS and going back to when you were an inner cell mass stage. And then your cells become embryonic stem cells in essence. And then from that, you can start using decades of history of developmental biology to turn them into different tissues with the hope that one day they can make therapeutically relevant transplantation organs. That's a big field, and I'll touch a little bit on organoids. They have some drawbacks. Traditionally, making these pluripotent stem cells, you need to induce foreign genes into them. Regulators don't like that in general, but now there are chemical ways of inducing pluripotency, and this is now becoming a very established technical procedure where with high efficiency, we convert them into, or good efficiency, high would probably be an overstatement. So they are fast becoming the template for regenerative medicine in terms of using pluripotent stem cells. So what do we do with them? What can we do with these special cells now? Everyone's talking about them. So cellular therapies, that's mainly what I've been talking about. So there are therapies ongoing. The first one was trialed in Japan. You can make macular cells or retina pigment epithelium cells of the eye. And there's a clinical trial that's well advanced in Japan for injecting those cells into the back of the eye. Heart failure, this is not to make heart cells, but you make sheets of cells that actually, when you put them over the top of your cells, when you put them over the top of your infarct, seem to help the infarct resolve, not necessarily make cardiac tissue. Perhaps the most promising trial is in Parkinson's disease. So Parkinson's is a disease, a specific set of neurons, dopaminergic neurons in the ventral part of your brain. And this has great efficacy because we've known that fetal transplants of human fetal dopaminergic neurons are effective treatments for Parkinson's, but we've never had enough and we've never had matched. So now we know how to make dopaminergic neurons in the dish using these cells. It's probably the most, and we know if we get them in the right place, they should work. So it's probably on the soundest grounds for a pluripotent stem cell making dopaminergic neuron human transplant. And there's a multi-center trial ongoing around the world. I think even Melbourne, there's some parts of that happening. And the great hope is being placed on this. And there are many other cell types that are being developed for treatment. What we can actually do now, because we know a lot about how organs are made in the embryo, because people like me over time have been studying their favorite organ system. Me, it's muscle, some make kidneys. Melissa Little down the road studies kidneys. We all know how they're made. And if we press the right buttons or activate the right genes, we can turn these cells into little replicas of human organs in a dish. And mostly they're not very good for transplanting. If you've got kidney failure, we can't make a full-grown kidney at this point and transplant it, but it can model human disease very well, and we can run treatments past them. So, can we alter the biology of a kidney organoid with a drug in a disease setting that we can use for a patient, say, with kidney failure? The ultimate goal with organoid production is to get them functional enough that they will be transplants, like pancreatic islets, for instance, are probably the most logical one. Because they're a small contained organ with a few cell types that should transplant and potentially help us regulate insulin production in type 1 diabetics. As I said, disease modeling is very important. We can make very pure populations of individual cells, heart cells, skeletal muscle cells, neurons. If you make them from patients that have disease in that particular thing, you can then take a huge bank of drugs. There are cancer organoids, so you can make organoids from gut cells that get cancer, and say which of my drugs will, if you have the primary, you can make a gut cancer organoid and treat it with drugs and work out which one works best for the patient and give that to the patient. So they're becoming extremely useful in what we call personalized medicine approaches, where we can make little avatars of your organs, and if you've got a disease, and try and test things for that.
Peter,
yep.
Are all these organs or organoids grown in synthetic media, or is there still a reliance on using, fetal calf serum and other sorts of media?
It varies from organ to organ. Mostly they are because they wouldn't be able to do so if they were heading for transplant. So all of these cells that are heading for transplant are made in completely synthetic organ tissue. You can't use fetal anything from an animal in that setting. Mostly they do use fetal calf serum in most of the settings here. But there's a big push to move it away from using fetal calf serum in many structures. And one of the fascinating things I haven't talked about, because it's not disease facing, is the use of stem cells in making food. And so, making meat or making fat. And there, of course, ethically, you can't use fetal calf serum because those cells will lose their resonance for the community that would probably use them to food. So yeah, some do, some don't. One day, all of them will have to be off fetal calf serum. So everyone's trying to do that, yeah. So in the disease modeling, I said you can use your own cells and make them into little mini organoids. It's a bit like mini-me in The Spy Who Loved Me. No, Spy Who Something. And drug screening I've talked about. They're very, very important with that, to screen drugs that could prevent or treat diseases which are modeled by these organoids. However, one of the real issues we have is taking it to the next level. So obviously, these are all pictures on the top here of different organoids, brains, liver, and lung. And they all, I can tell you, they all look like little organs. That looks like a little cerebellum of a human brain. But one of the great gulfs we have is that we know a lot how to make organs in the first place, like from developmental biology. So here's a brain of a mouse. We know how to make all these structures. We can know the genes and networks, and we know a huge amount about the adult condition. So this is a picture of a demented brain in the archives of the Wellcome Trust, because we're endlessly fascinated by our own health. So we study the homeostatic adult condition, but we don't know a lot about what goes in between those two conditions. So how do you grow organs and tissues to scale? How does that process actually happen in the animal? And I think this is a big gap, and there's a lot of interest in this area now. It's clearly, this is not how we make organs. We make a small organ and we make it bigger. We don't add bits onto it. And being able to recapitulate that in the dish now is the real frontier. How do you get enough oxygen into a big tissue in a dish without a vasculature? You have to grow a vasculature. Well, how does that happen? How do you get neurons in to a skeletal muscle organoid if you want it to contract? Because you need that to mature the cells. So these are the frontiers we're at right now. We have to understand how to make these small rudiments and grow them and develop them. But that is happening. And here's a beautiful picture of a colon in a dish, for instance. And these are used directly for looking at formation of the organ system, but also cancer modeling. And so this is perhaps the colon and gut are perhaps one of the more sophisticated areas, because it's a relatively simple tissue that was studied very early on in organoids, because you didn't actually need the embryonic stem cells. There are gut cells. So people have been making gut organoids for a long time. So yeah, this is what we try to do. I've sort of talked about this already, disease modeling, drug testing. So the idea is that we take your skin, so something's wrong with you, you've diagnosed a genetic condition, or your child has a genetic condition. We take a skin cell from the patient's body, we make pluripotent stem cells, and we differentiate that into a little organoid of the organ that's wrong in your body, and we can do disease modeling for your particular disease, or we can test specific drugs. And ultimately, we hope to be replacing the failing organ when we get sophisticated enough to do that. So that is the potential of organoids derived from embryonic stem cells from your own body through iPSC technology. But there are some fascinating things to think about when you make little organs in a dish. And perhaps the most challenging one to think about is a brain. I like to think of the movie The Man with Two Brains, which is one of my favorite movies of all time. Steve Martin, extremely funny movie, extremely funny man. And he was left with the fact that he had a brain, he had two brains, and didn't quite know how to make that situation work for him. With brain organoids, it's very interesting. So it's got to the point of sophistication now that I don't know, I haven't done this. My colleagues have made organs in a dish that are so sophisticated that they produce synaptic oscillatory waves in the cortex that are fairly identical to preterm human babies. And so when you think about that, that's pretty challenging. So are these organs in the dish actually thinking? And if they're thinking, what are they thinking?
They're thinking about playing Pong, aren't they?
Exactly. They're thinking, how the hell do I get out of here? I would imagine. But so, yeah, so when should we stop growing neural tissues in the dish? And how are we going to monitor that? And where is the frontier? I'll talk a little bit about some other ethically challenging issues that we're using for stem cells as we move on. So last year, and at Monash University, we had at Army and the Biodiscovery Institute, we had what I think is one of the most seminal discoveries in stem cell biology happen. This guy, Jose Polo, and his team, he generated something called an iBlastoid. So what I've told you about is tissues, individual, tissues you can grow in a dish. So everyone can understand that. You can grow a bit of muscle, you can grow a bit of brain, and we can study it. But what if you could grow an entire human being from an embryonic stem cell? This is not what they set out to do, actually. But they were interested in understanding how the signals that regulate differentiation of the iPSC is controlled. And it was a very simple thing. And they took some of these cells that had the Yamanaka factors in them and turned them back in time into the inner cell mass. And then what they did was they simply clumped them together. So these are the reprogramming factors, which trigger them to go back in time and to be embryonic stem cells. And then what they did is they simply aggregated them together, and actually it was physics, really. They brought them together in this wedge-shaped tissue culture well, and they let them differentiate it. And then they started taking images of them. And what they realized was that when I looked at these cells, they looked just like human embryos. So here in yellow is an antibody that recognizes a marker of the inner cell mass, and blue is trophectoderm. And when they took these cells and molecularly characterized them, they were identical to cells of human embryos pre-conception at this stage of the development. They call them iBlastoids. They don't have some of the lineages that would allow them to implant, so they'll never make a human being. But to be frank, this is very close to a human embryo. And all they needed is one skin cell. And that's all they needed. So how far should we go with this particular technology? How long should these cells be allowed to develop? And in fact, this issue was so perplexing for regulators that Jose's work was halted immediately. And he kept telling the regulators where he was at, and they told him how far he could go. But we still don't have, from the NHMRC, an ethical framework for the growing of iBlastoids, even though other jurisdictions have it. So this is a perplexing question, even for government and regulators at this point in time. So technically, you no longer need a male and a female. You certainly need a womb. We have to implant somewhere. So we need women, but we actually probably don't need men anymore. It's a long way to go before that becomes a human embryo, to be quite frank, but just thought projecting into the future. If we can make human embryos, this will be a clone of yourself too, of course. That's another ethical thing. Do we want, what's that movie where they clone people for organ parts? I can't even remember what that was called. Anyway, all these things are not, thought projecting 20, 30 years. These are technologies that you could postulate as a futurist might be on the horizon. Now, this is really interesting because we could study the pre-implantation human embryo. We can start to, work out what goes wrong when things don't implant and all these things. We have great models for that now. So it does have direct benefit to human health, but it's also ethically challenging. Another really fascinating area that this doesn't happen in my research institute, but I'm really fascinated by xenotransplantation. So this does go against the flow of public opinion where, animal organs shouldn't be harvested for human use. But, it's something that's long been postulated. Here's Jean-Baptiste Denis doing one of the first xenotransplantation. and he's actually doing a blood transplant between this poor lamb and an even poor suffering from a lamb to a man. That's crazy stuff. But anyway, can animal-human chimeras, can they be used for solving some of our medical problems? So this is a real frontier and of interest. We know that when we make chimeras between animal pluripotent stem cells and different species, they will form embryos of a certain degree. And can we make organs from one species, i.e. us, in an animal? Because currently we can't. I've just told you the problem with organoids is we don't know how to grow them, but every animal knows how to grow an organ because that's programmed into their development. And if we just provide human cells to grow in that animal in the right place, we'll have an organ of approximation of function. And maybe that's something we can use.
Peter, I'm told that the movie was The Island. Is that the one you're thinking of? Yeah.
It sounds about right. Yeah. You can see I'm not so good on popular culture. I usually rely on my daughters to inform me of the movies we've watched together. But yeah, so that's exactly right. Yeah. So yeah, this is kind of a thing that's going on. So pigs are the animal of choice, our beloved friends, and mainly because the organs of pigs are roughly the same size as us. So people have thought about this for ages and wondered if pig hearts could, in particular, and I'll show you why. And there have been problems because there are several, they've rejected basically pretty rapidly pig hearts as transplants. But this colleague, David Cooper in the University of Pittsburgh, have been working out which of the cell surface proteins on cells are actually causing that rejection in the pig heart. And because now we're long into the age of genetic manipulation of many animals, we're able to knock all these genes out in all these animals now, where before were never able to do this. So theoretically speaking, a lot of the pig tissues could be used because they function in a similar way. Obviously, you'd always be immune compromised and suppressed, but could it work? So actually, there was a really quite astounding study where they took a genetically modified pig heart and put it into a baboon, and this baboon lasted for nearly a year. It was extraordinary. It was very, very heavily immunosuppressed. So technically speaking, you know, these things can happen. And this is the first time this has ever happened, that a pig heart into a baboon would last more than 6 months. So they worked out that it can be done. So that was in 2018. And then we know time has moved on because we've just had the first pig to human heart transplant that happened last year. This was a very late stage heart failure patient. And my colleagues at the Victor Chang, where I used to work, will tell me that these poor people have very, you know, even with transplantation, have very poor prognosis with a human matched donor. So he lasted for several weeks, I think a month or 2 months, and he died. But the heart worked perfectly well and the time that it was there. So it was believed that it was the comorbidities that took him out in the end. So this is an interesting area. But perhaps more interesting is this idea called plasticist complementation. So this is the stem cell part coming back in now. So if we can, as I said, if we can trick human cells to make the organ of interest in your pig, then you won't have any rejection problem. I mean, obviously this is a long-term project if you want your own cells, because to grow a piglet to term with your cells in it might take 3 years, but if you know you have a dodgy heart, maybe you can get it done. But this is, again, a thought experiment. Well, some of it's a thought experiment. And people are doing this experiment, but the actual therapy part is a thought experiment. So the idea is that you inject human cells into this pig embryo, and you hope that it makes an organ that's mostly made of human cells, and then you can transplant that organ into yourself when you have organ failure. So this is the dream of Hiromitsu, who's been circling the globe trying to make this happen. So originally in Japan, he was going to do this, and he was going to make this human-animal hybrid and bring it to term. And finally, after years of planning, he thought he was going to do that, but then Japan changed the rules, so they wouldn't allow him to do these human pig chimera experiments. The idea about that is a tricky one, because what if you get the cells in the wrong place and the pig starts watching TV and voting for Donald Trump? You're in trouble. So, this is sort of the unrealistic fear that regulators have. So then he moved to California because the rules seem more liberal there. And by the time he got and set up his lab, California changed the rules. So he's never been able to actually do this experiment. But nevertheless, the science is moving on. So yeah, can we grow a human pancreas in a rat, say? Could we make human lungs? And so what think is to try and get around this issue of cells seeding everywhere in every organ. We've tried to work this out, because this is obviously the challenge we don't want to have, right? You don't want to wake up or bring to term something that looks like this, which is ridiculous, by the way. But anyway, it makes the point there are concerns about it. So basically, what they've decided to try and do, in essence, to do this is knock out genes that are essential for development of an organ in a pig. So say you need gene X for making a pancreas, and you knock that in the pig and it can't make its own pancreas. And you take the human cells, the only cells that are ever going to be in that pancreas are human cells, because they haven't got that gene knocked out. And so this is called blastocyst complementation. And they've done this with a number of things, and they show actually when that happens, there's very few cells that populate any other tissue other than the one where it's going to be made entirely by human cells. But to get around that too, they're going to knock out genes in the human cells. That's what they think they'll try and do, that can make neurons, because that seems to be the main problem that people have. You don't want to have a thinking pig. Although to me, they seem very intelligent animals in their own right. So a few scattered human cells, I wonder, would make them dumber, actually. But nevertheless, this is a really interesting, rapidly moving field, and one that may, in the short term, get around our inability to grow organs to term in organoid dishes, organoids in a dish. Okay, so that's what we're doing with pluripotent human stem cells. And I'll move into a field that's way less controversial, because we mostly have these cells, or we want to try and create them, tissue-resident adult stem cells. So here I'm on safer ground because I actually work on these guys. So those of you who've been to the gym or have had a soft tissue injury know that your skeletal muscle can repair itself. And this is all down to the existence of these tiny little guys called satellite cells, which are skeletal muscle stem cells that reside on the outside of the muscle fiber here. They're called satellite cells simply because they're on the outside of the fibers. So if you go to the gym, mostly, when you go to the gym, you're doing something called hypertrophy, which is making the existing muscles grow bigger. But occasionally, if you go crazy and you really lift heavy weights, you'll tear your muscles, and then you'll activate the repair pathway, which is the stem cell mediated process. They're very active in disease, such as muscular dystrophy, and they're believed, at least in part, to underlie the wasting that happens with aging. So sarcopenia, we're all going to get it. Men over the age of 25, I'm sorry, it's already happening. You're going to lose up to 60% of your muscle mass by the time you get to 80 years of age. So get thee to the gym is the answer to that. And stem cells are in part behind that. So, but I like this as a cautionary tale because they're very simple. They're one stem cell on the outside of a muscle fiber and only makes muscle. But yet, unlike the hematopoietic stem cell, which has been, skeletal muscle stem cells are the second oldest stem cell known to man. They were discovered in 1961, a decade after the hematopoietic stem cell, and they're a simpler system. So you think we might have been able to do something with them to treat muscle disease by now, but no, we can't. When we take them out of the body, they poop out. And when we put them into another body, they don't do what they're supposed to do. So we don't really understand the signals that turn them on. We don't understand the signals that need to come with them to generate them. We don't understand how they undergo self-renewal. And, I think this is a really intriguing example because it's very simple and we should be able to harness it, yet we haven't. So that means it's more complicated than we could possibly believe. And sometimes you just have to be a bit patient with the knowledge of how we do things. Okay, so that's some caveats. There are we can stimulate endogenous stem cell systems with different processes. Immune modulation is one thing that works very well. The immune system stimulates stem cell compartments directly. That's some of the work we've been doing. It's not that we can't use them in vivo, we just have trouble taking them out and putting them back in. So there are systems that we use to turn them on. Okay, regeneration in biology and regenerating systems. So I've talked about endogenous stem cells, and that's one form of regeneration. But really what we really want to do is mimic the invertebrate system. So we all know, some of us as children have gone down to the beach and pulled an arm off a starfish, maybe come to the pond a week later and realized it can grow back. Few of us have diced up a planarian. I'm one of the people that has, and this is a poor planarian that you can dice into these tiny little pieces, and this has actually happened, and it makes a head and a tail from each of the little pieces and regrows. And this is actually stem cell based. And actually you can fuse two head pieces together and make a pushy-pulley planarian, which is rather cruel, I think. They don't know whether they're coming or going, but nevertheless, they regenerate perfectly fine. So my former boss, Nadia Rosenthal, was very interested in newt and limb regeneration, and she's just going to tell you a little bit about this champion of regeneration. So axolotls and newts have this amazing ability to regenerate a lot of their organ systems, including their limbs, which is, a really amazing attribute and fairly unique amongst tetrapods. So she's just going to tell us a little bit about that. No, she's not. No, this is the one without sound. She's going to do the zebrafish one. I have to do this one. Okay, here's an axolotl where the limb has been resected, and it grows back to produce a perfectly normal limb. So how does it do this? Well, it has all the tissues, bones and nerves exactly the same to us. And what happens is that when the steady hand of the surgeon comes in and resects the axolotl's limb, it immediately heals over and forms a tissue called a blastema. And in the blastema, what happens is something called dedifferentiation of cells. This is a very unusual trick in regeneration, and they form sort of individual tissue stem cells that occur within the blastema. Just like the Lernian hydra, it now can regenerate. That's mythical, by the way. It's not real regeneration. Please don't lop heads off anything when you go home, least of all your partner. And you'll see that these individual tissue dedifferentiating can divide and regenerate and regrow the whole limb. So really, this is something quite remarkable. So we study them in the lab to try and understand what are the cues and triggers that allow within a tissue for this regeneration to happen. Really amazing if we could take some newt juice and paste it over the limbs and regrow limbs from people who come back from war. Actually the Department of Defense in the US are very interested in axolotls and newts, because now with these amazing Kevlar body armors, the most injuries that come back are loss of limb or limb incapacitation. And they're left with an incapacitated person the entirety of their life. So again, this is a bit future looking and a bit sci-fi, but we do know some of the signals and cues that trigger pro-regenerative functions in the limb of these animals now, and they can be manipulated at least partially to achieve better regeneration in non-regenerating systems like mammals. My favorite regenerating system is the zebrafish. You probably look at these humble little fish and think, what can we possibly learn from these guys? But they can regenerate most of their tissues and organs. And most of the genes that they have are the same as ours, and most of the cell types that we have in our bodies are the same as them. Basically, we're just modified fish. It's a sobering thought. So zebrafish can regenerate their hearts. We have 300,000 people every year who are in heart failure. Some of those will be on the transplant list. We only have 80 transplants to offer every year. So this is a pretty big problem to solve. So if we could work out how to regenerate cardiac tissue, this would solve one of the greatest morbidities in the Western world. So a lot of people are studying regenerating systems that are unable to do that, to try and work out how they can do it, how they can implement them in settings where it doesn't happen, such as our cells. surprisingly and amazingly, if you take, mammals can't regenerate. They form this permanent scar. If you cause an injury or an infarct where the blood vessel is blocked to your heart and your cardiomyocytes can no longer survive, they die and form a scar. Both newt and zebrafish have this amazing ability to regenerate, and zebrafish is the most efficient. This is the ventricle of the heart in zebrafish. If you cut off a third of the ventricle, don't do this at home. Cut off a third of the ventricle of a zebrafish heart. 60 days later, it grows back and it's completely normal. I think that's remarkable. That something can even function with 30% of their heart gone is quite remarkable, but it regenerates it completely. So Nadia now will tell you how that happens.
Zebrafish, which any of you who are fish fanciers keep in the fish tank, can get to be about 2 inches long. It's got a little heart with just 2 chambers, 1 atrium and 1 ventricle, and that pumps blood throughout the whole body past the gills, which are the equivalent of the lung in a fish. The heart is very muscular, just like our heart. Inside, there are very thick muscular walls that allow the heart to effectively pump throughout this very complex series of veins. Now, let's see what happens if we cut off the tip of the heart. Ouch. Oh, don't worry. The fish is okay. It's okay. The fish immediately closes up that wound. Within seconds, the clotting process starts up. Now, in our heart, what would happen is there'd be a big clot, and eventually that clot would essentially heal over, but nothing more would happen. But Ken Poss and his co-workers have recently noted that aside from the fact that there are cells within the zebrafish myocardium in the red muscle that can actually activate like satellite cells and start to divide to make new muscle, there's also this slowly proliferating, engulfing layer of single cells on the outside of the heart called the epicardium, which is really a magic layer of cells because it engulfs the whole scar area. And then in a really remarkable series of events that recapitulate developmental biology of the heart, a series of growth factors, in this case, a growth factor called fibroblast growth factor, is produced by the heart as it's regenerating. And this fibroblast growth factor, or FGF, docks into epicardial cells set on the edge of the heart. And those epicardial cells, with this signal, know to march into the myocardium. So there they are, marching in. Now, the next thing they do, obviously now we're in a macroscopic picture, is a very important, absolutely fundamental part of making new muscle, which is to vascularize it. So with the new veins in there, the fish ends up with a brand new heart with new muscle, and not only new muscle, but all of the appropriate coronary vasculature to innervate it. And the fish can actually survive and swim away. Zebrafish, which...
That's enough from Nadia. Yeah, so thank you, Nadia, for that. She's a cardiac regeneration expert, so always good to hear from an expert. And, that's an amazing party trick that we would love to have. So a lot of people around the globe are studying that fact. In fact, even big drug companies have cottoned onto the fact that zebrafish can regenerate their hearts and we can't. And their solution is for you to buy their statin, which I don't think is as good as being able to regenerate. But there you have it. Right, so the final thing I want to mention in just a few slides on this is tissue engineering. That's where we take the ability to make scaffolds and artificial polymers and combine them with cells to try and craft them into tissues or structures we can use to implant into the human body. So this, I think, is a fascinating area because it uses all this deep knowledge in natural and synthetic polymers that can instruct cells to do things in a particular way, can deliver drugs with cells, and it gives us more control over the implanted cell, which is ultimately what we want to do. So you can make synthetic polymers. You can apply those polymers with regulatory signals like growth factors. You can put the cells into these synthetic polymers, and now we can do 3D printing using these, what they call bioinks. So we're trying to make a 3D imprinted muscle that has a particular shape and structure that we're going to put muscle cells in, we're going to put vascular cells in, we're going to put nerves in. we're going to try and see if we can make an organ-typic 3D printed organ that we can do disease modeling, but also we hope one day, say in situations such as volumetric muscle loss, when you lose so much of your muscle, it can't regenerate anymore. These could be used to provide the types of structures that will regenerate tissue fully. So I think this is a fascinating area. And for instance, one of the examples we had recently is we used the fact that zebrafish muscle regeneration occurs very rapidly and very well. We worked out what the molecules were. It was a component of the immune system that was doing that. We found the signal. And then we worked with our colleagues to create a biomaterial with fibrin, a particular type of fibrin. And we said, well, this fish molecule, let's see if it'll work in the mouse. And this is this volumetric muscle loss that I've talked to you about in a mouse. If you cut out enough of the muscle, in this case of the quadricep muscle, it won't regenerate. But here in a tissue biomaterial with our molecule put in place, it without it, you don't get any regeneration. You just get the blue stuff, which is fibrosis. But when you put this molecule in a biomaterial, which we know stimulates the stem cell compartment, we get remarkable regeneration from that setting and no fibrosis. So I think this is a real frontier future where we take our knowledge of not only tissue homeostasis and repair by stem cells, growth factor biology, combining it with tissue engineering, delivering a really potential industry-based molecule. And we've now started our own company based on this molecule, trying to deliver this in both tissue regeneration scenarios like volumetric muscle loss or surgical muscle weakness, or also dystrophies where we think it will be useful. So I'm just going to end with my friend Prometheus, just bringing back the classical theme to the end. As I said, it was often thought regeneration was a punishment. Either you were inflicted by an animal or was inflicted on you. But I hope I've convinced you that perhaps regeneration can begin to be harnessed. We won't have to be chained to a rock and eaten by an eagle all the time in order to experience. We might be able to go down to the clinic, buy a set of cells that are matched to us, or give our cells, make a tissue, and one day that can be a path to regeneration. And if you want to know anything more about regeneration, or you're interested, this is ARMY's website, and I'm always happy to spend some time talking to people about the potential of this amazing science. And I appreciate this is dinner time, and listening to science can sometimes be super boring, but I hope we got something out of that. So I'm happy to take any questions you have, or don't, if you have any, just fire away, basically.
Well, so I've got one to begin with, and that's that you mentioned that the chemical induced IPSs are being done. It's being done successfully now. I remember a few years ago there was a lot of controversy about whether that was real or not. But from what you say, that's the problems have now been solved.
Yes, that was the STAP controversy, which was really controversial, and that was just clear fabrication. And no, they have managed to, its efficiency is nowhere near as good, but there are chemical induced pluripotency procedures now.
Yeah. I haven't got any questions in the chat box, just comments saying fantastic talk, terrific talk, Peter. Thank you very much. So unless some questions appear, I think we'll just remains us to thank you for a very interesting and entertaining talk, Peter. And here's a question, last one. And the question is the risk of cancer raised in regeneration?
Yeah, that's a very good question. And it's one of the ones that does occupy the regulator's mind all the time, because cancers have stem cells, cancer stem cells, and cancer is simply unregulated differentiation or proliferation of an organ. And it is possible that stem cells could have this, could be transformed if they're highly proliferative cells. So there are a lot of technologies being developed where they have fail-safe systems involved, such that if you say you implant your IPS-derived tissue or organ, and you didn't quite get all the cells to differentiation, and there is a highly dividing population that does look like it's going to go cancerous, you can actually kill all the cells.
So you sort of put in an Achilles heel, do you?
You put in a Trojan horse into your
Yep.
cell and you add a chemical to it, a harmless chemical normally, and it will kill all the cells that you've transplanted. So they're that's called fail-safe systems. So my colleague Andras Nagy at the Institute is developing such systems. But yeah, that is a very good question and a real concern.